Hi guys, we're back on cellular respiration, a lesson on anaerobic respiration. So here's our dot point, and our dot point says that we need to know the location of anaerobic cellular respiration, its inputs, and the differences in outputs between animals and yeasts, including ATP yield. So we're going to look at two different types of anaerobic respiration, and here we go. Before we start, I just want us to go over the types of reasons that we use ATP. Remember, ATP is that fuel for all of our cellular processes. So we can use it for all of these things, and I'm not going to go through them again. And then let's just go through the process of glycolysis really quickly. You can see, what do we put in? We put in a glucose molecule, we get out 2-pyruvate. Yeah, we're going to ignore the water output here. We get out ATP and we get out this NADH. And so from glycolysis, how many ATP do we get? So remember, if we go back to that dot point, it says, what is the ATP yield? At the moment, we've got this, okay? Now, the reason I'm going through glycolysis again is because glycolysis doesn't involve the need for oxygen. When we think about the word anaerobic, anaerobic means without the need for oxygen. So at the moment, we can generate 2 ATP from our cells, or from any cell, without the use of oxygen, using the process of glycolysis. Okay, now this is great. We don't need oxygen. Let's get some energy. Not a lot of energy, but we've got some. Now, there is a problem. Okay, we can't just use this all the time. What will happen is we'll get this build-up of pyruvate, and we'll get a build-up of NADH. Okay, as they start to build up, their concentration increases. So normally their concentration would be, so here's the concentration gradient like this. So normally they just get used up constantly, which keeps this concentration low. But when we aren't making them, when this is not happening, what happens is their concentration keeps building up and up and up, and eventually lowers that concentration gradient and stops the reaction. So when it stops the reaction, what's going to happen? We stop the reaction, we get no ATP formed. If we don't get any ATP formed, all these things aren't going to happen. So this is when we as animals do a process, and the other one is yeast, and that's the experiment that you're going to be doing too. But we'll start with anaerobic respiration in animals. Okay, so we're going to start here. This is number one, is we're going to look at animals and the production of lactic acid. So, oh, cute puppy. Yeah, so here is the production of lactic acid. This is glycolysis. So this whole stage here, that top bit, that's all glycolysis. Okay. So glycolysis is there, and glycolysis makes us 2-pyruvate. It makes us, get rid of that because that's wrong, 2 NADH and 2 ATP. All right, when we do that, we want to be able to get rid of, we need to get rid of these. Okay, if we don't have oxygen, we need to get rid of these products. So what our bodies do as animals is we turn them into, sorry guys, we turn them into lactic acid. So this is in animals. Now, there's a very good video that I'm going to link to here. So this is the video. It's called The Crawl. I want you to click on it, have a watch, and then come back. So overall, what does our anaerobic respiration look like in animals? We need some inputs and we need some outputs. So we put in some glucose, which is what we've got here. So we've got over here, you can see the input is glucose. The other thing that we need to put in is we're going to put in these two things. So we're going to put in glucose, and we're going to put in this 2 ADP plus PI. What do we actually get out of this scenario? So our outputs, I'm going to do in orange. I'm just going to get rid of some of these pen markings. So there are our inputs. Our overall outputs in this scenario are going to be lactic acid, and the ATP. The reason I'm not mentioning any of these bits in the middle here is because that pyruvate is used up, the NADH is returned over here. So when we're summarizing anaerobic respiration in animals, we can do that just by saying that we put in glucose, we get out lactic acid, and we get 2 ATP overall. So I'm going to stop the video there and we'll go on to the next one.